There we go. So today is the last lesson about biotechnology. What we will do today is we will start with the quiz, and then we will watch the video together of the activity you have to do, and the rest of the time is for you to do the activity. You can do this with somebody who you like. So I'm not going to divide you in groups. But you can work with one or two persons together if you would like. If you would like better to do it alone, you can do it alone. Okay, so let's first do the quiz. The quiz which you have to do, you can find it here in Canvas under week number 9, Genomics and Biotechnology, and you do Genomic Review Questionnaire. So that's the one you have to do today. And then the other thing we're going to do today is this genomics and biotechnology activity. I figured it's better that we do an activity about it than that I explain another lesson all the time, me talking. It's better to do it a little bit more dynamic and that you do the activity to discover more about this topic. So please start, and I will monitor how you're doing.
Mister, how many attempts we have on the quiz?
The quizzes are usually only one attempt, Arturo. When I give you two attempts, this was an exception. So a quiz usually is only one attempt.
And we
So
can
for
do it
this,
in Teams?
the quiz, no.
Ah.
No, the activity
Okay.
after the quiz, yes. But now you have to put your camera on and you have to do the quiz as good as possible. Please could put your cameras on, and please keep in mind that next week it's the let me confirm that date exactly in the meanwhile you do the quiz. The date of the second partial, to be absolutely sure that it's next week Thursday. I wrote down the 15th, but I just want to double check.
Mister, what is the name of the quiz?
Genomics Review Questionnaire.
Okay, thanks. Mister, I don't know if it is a mistake or am I wrong, but in question number 2, it appears instead of adenine, adenive.
With a D, yeah, I changed that, but that's so weird about Canvas. I have seen the mistake. It says adenibe or adenide instead of adenine.
Adenive.
Yeah, it's meant to be adenine, but supposedly I changed it, and it always appears with the mistake. So yeah, you can click on it. Just ignore the spelling error. And then if that mistake is still there, there is another mistake that you have to order the DNA parts from smallest to largest. This has to be the opposite. It says largest to smallest. It should be smallest to largest.
Okay, thanks.
Yeah, so the smallest thing first, if you get that question, because there was also a mistake in that one. I corrected it, but Canvas, for one kind of reason, it doesn't register the changes I do. And that's a problem that I already reported, but I think they cannot change it.
Mr. What was the name of the quiz?
Um, Genomics Review Questionnaire.
Okay.
So, teacher,
Yes?
adenine it's an adenine,
Yeah, no,
like in the quiz.
yeah, exact. No, it's wrong in the quiz, but I changed it with an N, but it didn't register the change. So just suppose there is no spelling error, okay?
Okay, okay.
Okay, here it says to place the molecules from smallest to largest. So we place them from largest to smallest?
No, 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 that it should be smallest to largest, I think. A ver. Yeah, from smallest to largest. That's what this one did change. That's weird. Yeah, because it said largest to smallest, and it should be smallest to largest. If you have this question, guys, I also want to say that there is something confusing about it. Okay, the correct answer is that DNA is smaller than the gene, but it really depends what you're looking at. If you're looking at the DNA helix, yes, this is smaller than a gene because a gene is a whole piece of DNA. So it really depends how you see it. But that's why I tell you, from if you have from smallest to largest, you put DNA first and then gene, okay? Because a gene is a lot of DNA together. But if you take the whole DNA of an organism, it's more than a gene, of course. So it's very relative. This I found it a bit confusing, so that's why I tell you that you put gene in the end. Gene is the largest, okay? And the base and the nucleotides, I explained that. You should know which is bigger. So just to confirm, guys, indeed, the second partial is on the 15th of October, okay? Next week, Thursday, okay? And of course, what you have to study for the second partial is very simple. It's everything we have seen so far, from the beginning until now.
Teacher, by everything, it's also what we saw in the first partial or just the
Yes.
second one?
In Tec de Monterrey, sciences is always accumulative. So it means that you always have to also know what is from first partial. Of course, the exam will be focused on second partial, but you're supposed to know what the things from first partial do, because the final exam is of everything also. So it's in your it's in your interest to review everything again, because otherwise it will be a lot when the final exam is there. So I don't know how that is with other subjects, but in Tec de Monterrey, according this is I mean I've been the director of sciences before. Now it's Gerardo Alvarado, but the instruction of Tec de Monterrey in any science, in any mathematicas in Tec de Monterrey, the exams are accumulative, which means that partial 2 is partial 1 and partial 2. You know that you have to study everything that you know until now. Okay. You cannot see the partial as a quiz that you only study the little segment you have seen so far. No, the partial is everything from the beginning of the course until now. And the final exam is everything from the beginning until the end, okay? And that's why I think it's good that it's accumulative, because otherwise you would, yeah, you study the things more, you know, and we see that by doing this, students often have better grades as well, because they repeat it more often. And the other advantage is that if you study in this way, you retain the information better, and it will be easier for the final exam as well. So you will get questions of first partial too. They all come from the same database, so there might be a question from the first partial. But you can do it, guys. I have complete confidence in you. But take into account that so there is students that do the second partial better than the first partial, but there is also students that do the second partial worse than the first partial. It really depends. But there's always students that do a lot better the second partial. Because they know how the questions are asked, they study more in detail, they know how to study, so you have that advantage. Okay, let's see how you're doing with the quiz. And you're doing great, eh? In fact, everybody can still pass my course, and normally I have at least 7 or 8 people that fail the course, and this year with the online courses, which are a little bit more light, it looks really well. I think you're doing well. You're doing well, and the other group is even doing better. With all respect, but you have good grades, but the other group is really strong, very, very strong. And you are already good. Okay, I see one person finished, so we still wait a bit, okay?
Papi, ¿vas a tener 5 clases?
Three.
¿Pues? ¿Y ya terminaste las clases? ¿Tienes que hacer muchas clases?
Mm.
Mr. Remy, you can only do one attempt?
Yes, only one. It's been, in fact, an interesting week because we are in week 9 and there is 16 weeks. So it means that we're already over half the course. So we're, in fact, we already past the 50% mark. So we're even with week 9 finished, we're in 55% of the course, more or less. So we still have, next week is week 10, so we still have 7 weeks to go. So we have week 10 until week 16, and that's it. 7 more weeks, and there is just like one holiday there in November, I believe, but I don't think it's for us, right? It's the 16th of November, Monday now. So 7 weeks, that's less than 2 months. It's 1.5 months and it's already Christmas. I don't know about you guys, but I go camping with Christmas. Yeah, I feel like camping. Nobody does that with Christmas, but we do. Okay, are you done, guys? You should be done more or less, huh? Because I give you 15 minutes. It was not so hard. I see some good grades. I see some not so good grades, but overall it's okay. So then what we're going to do is watch the video together about the genetic modified mosquitoes. But before we do that, I don't know if you heard in the news, but it was Nobel Prize this week. And the Nobel Prize for chemistry, which is really not chemistry, it's more biochemistry or biotechnology, it goes to Emmanuel Charpentier and Jennifer Doudna. These two persons, they did really mind-boggling work because what they did is they invented the method of the Cas9 and the CRISP gene. I don't know if you've heard of that, CRISPR and Cas9 genetic editing. So why is this so important? Well, these women, what they invented, they invented like a tool, like a kind of enzyme, a gene, let's say, and that is able to be programmed, and you can get any gene you want. So you say, get me this gene and cut it out of the genome and separate it. So this is very important to genetically modify organisms. And I thought this was interesting news because we're going to see the movie now. We're doing genetic modification more and more. For sure, you have heard of Monsanto, or you know, soybeans, strawberries, cotton. In fact, a lot of the GMOs, you have heard of GMO, are genetic modified organisms. And especially in agriculture, we have a lot of that. The corn you eat, it's corn that makes its own pesticide, and it cannot go bad. So there is no weeds growing in the fields. And then we have modified soybeans that become bigger. We have genetic modified strawberries that do not freeze at night and they are redder. So we are eating a lot of genetic modified organisms. I think this is something that I don't really like, because I don't think we should really engineer our food in such a way. Moreover, what I believe is really bad is that Monsanto made suicide seeds. Have you heard of suicide seeds? Just to make more money, they make the organisms that they don't produce, they make the plants in such a way that they don't produce any seeds anymore, so that the farmers have to buy the seeds from the company. So this is really, I don't agree with this. I do think that genetic modifying, we have done it all in all history of mankind, but not at levels that we do it right now. So there's a really big ethical issue here. Let's see about this video about genetic modified mosquitoes. It's 8 minutes, and you will do the activity about it, which you can find in Canvas. And then let's have a look what you think about this, okay? Because it's also really, I mean, is it correct thing that we are doing or not? It's really up for debate, let's say. Okay, the video, I'm gonna share my screen with you so we can watch it together. There you go, share the sound. And that's it, what we're gonna do. Friday always has to be a bit lighter because it's Friday. So enjoy this, guys, it's very interesting.
Brazil, late 2014. Doctors noticed a disturbing trend. An increasing number of babies were born with abnormally small heads, a condition known as microcephaly. The spike in microcephaly was linked to a virus called Zika that is spread by mosquitoes.
I just never dreamt, as an epidemiologist of infectious disease, that I would see a congenital transmission by a virus that would do such a great damage and would be transmitted by mosquitoes.
Throughout the Americas, mosquito bites are tied to many viral outbreaks. In the months before Zika spread in Brazil, chikungunya, a tropical virus that causes terrible joint pain, invaded the Western Hemisphere, infecting almost 1 million people. And West Nile virus sickened more than 2,000 victims in the US, with symptoms ranging from fever to paralysis. To stop these outbreaks, scientists have turned their attention to fighting the mosquitoes that carry these diseases. And one of the weapons they are using is genetic engineering. In South America, the Zika virus is spread by a mosquito called Aedes aegypti. Other diseases, including dengue, chikungunya, and yellow fever, are also spread by this persistent insect.
Aedes aegypti is the cockroach of mosquitoes. It lives indoors and outdoors. It bites daytime and nighttime. It develops resistance to insecticides easily. The eggs can overwinter for a year. To control it, we have to kill the adult mosquitoes, the larval mosquitoes, both inside and outside.
Health officials have used sprays and screens to protect people from diseases spread by mosquitoes. But since 2002, scientists at a biotech company called Oxitec have been investigating a more direct approach. a line of genetically modified Aedes aegypti mosquitoes called OX513A.
The goal of this procedure here at Oxitec is to use our mosquito, OX513A, to reduce the population numbers of the Aedes aegypti mosquito in the wild.
When released, these mosquitoes reproduce with wild mosquitoes and cause their offspring to die. Although there are concerns about this technology, authorities have allowed limited releases to study its efficacy and safety. So how were these genetically modified mosquitoes produced? The process involved engineering mosquitoes with 2 new genes.
What you do first is create or synthesize the DNA you want to insert. So in our case, we used the 2 genes that we inserted, the lethality gene and the fluorescent marker.
The genes were inserted in the genomes of mosquito eggs. The lethality gene makes the developing mosquitoes dependent on the antibiotic tetracycline, which is fed to them in the lab but is not available in the wild. The fluorescent marker gene produces a protein that glows red when exposed to light of a certain wavelength. It tells scientists which mosquitoes have the lethality gene. After the first genetically modified mosquitoes grew into adults, scientists bred them in the lab. These mosquitoes passed on the genetic modification to each new generation, giving rise to a colony of genetically modified mosquitoes, all dependent on tetracycline.
So this is our female room. You can walk in. In these big cages, what we have is our egg-producing colony. In total, about 12,000 females per cage. And they are here to produce the egg. So our genetic modification has been done in 2002. A stable inserted in the genome of every single of these mosquitoes, which are here in this room. They lay their eggs in these pieces of paper. All the black little dots are eggs that have been laid.
When scientists plan to release genetically modified mosquitoes into the wild, they collect eggs from this colony. They place the eggs in water, where they hatch into larvae and then grow into pupae. At this stage, technicians separate the males from the females by size.
These are a separate female, and that's a male pod.
The male pupae are significantly smaller than the females. Feeding them tetracycline, the antidote to the lethality gene, keeps the mosquitoes alive as they grow into adults.
So we give an antidote in the lab to the mosquito so that they don't die. And then we go release our male into the field.
Male mosquitoes don't bite people, so they will not spread disease, but they live long enough to mate with female mosquitoes.
And then life becomes exciting for them. They get to do what they're best at doing, which is searching females, copulating with those females. And our males will deliver the lethality gene to their children. And in the wild, the babies do not come in contact with the antidote. And there, they die.
Well, here in this van, we can fit almost 800,000 of male mosquitoes. As we go driving through the area, every time the app beeps, we just open one of the spots in here and the mosquitoes will fly around and do their job.
Once released, the genetically modified male mosquitoes mate with wild females. The male carries 2 copies of the genetic modification, so all the offspring inherit a copy of the lethality gene. The wild offspring will not feed on tetracycline, the antidote to the gene, and they will die before reaching maturity. Scientists monitor how the technique is working by capturing larvae and determining what percentage have the telltale red fluorescence. Studies have shown that with repeated distribution, the wild mosquito population of a village or neighborhood can be cut by 95%. If the technique continues to prove effective and safe to humans and wildlife, it could be applied to other species of mosquitoes and help stem the spread of many dangerous diseases.
So that's it. So what do you think of this, guys? It's like a positive story of how we genetically modify organisms. Everything seems to go well. So we're going to do an activity about this. Somebody has some comments about this? Who wants to add something about this?
Impressive. Like
Yeah, it's
how
very,
they do it. And
yeah, it's very interesting, and but it's a very good story, and indeed it seems to work. But what worries me is nature doesn't work like that, you know. Nature always has an answer for every reaction. There is an equal counter reaction, and we don't know what that counter reaction is, and I'm not sure if they thought really well about it. So I mean, it looks harmless, it looks okay, but always be critical about this. So the activity you have to do, guys, I told you, I'm gonna leave you super free in doing this. You can do it with 1 or 2 persons maximum. 2 extra, you know, like maximum at the total of 3 persons. But everybody has to hand it in individually. And if you work in group, put all the names, and it's basically a nice activity because it brings all the concepts a little bit in one tool, let's say. So here you first have to do a matching exercise. What is DNA? What is the gene? What is genetic? What is bioethics? And so on. So you do a matching. And then you have questions about this genetic modified mosquitoes. You have to answer some questions and check what are the advantages and disadvantages. So it's a nice exercise for you to do. So you don't have to stay here. You're free to disconnect to do that. But don't forget, you have to hand it in on, I don't know, I'm going to give you until next week, Wednesday. Yes, on next week, Wednesday. But if you can do it before, even better, because I have to check it before the second partial finishes, okay? If you want to stay here, you can stay here connected, but you don't have to, okay? I'm going to give you a little bit freedom to do this because we don't have to be connected all the time. So however you want, okay? Is everything clear, guys?
Yes, Mr. Thank you.
Yes, sir. Thanks.
Okay. So use the time to work on this. If you, for instance, have this next class, okay, I understand, but try to already work a little bit on it. You still have 20-25 minutes before your next class starts, and you could finish it 90% in this time, okay? So it would be nice if you use the time properly, okay? So for me, this is everything. I wish you a good weekend, and we see each other next week, Wednesday, okay?
Thank you, teacher. You too.
Thanks.
You're
Bye,
welcome.
teacher.
Bye-bye.
Thanks.
I